Over the last few videos, we've been talking about radioactivity and the radioactive isotopes, and there are many uses for this in modern society and in science. One of the most cool uses, in my opinion, is medical applications for radioactivity. And in addition to actual treatment, where you actually use radiation to kill the things that you want to kill, you can also actually detect the presence of something you need to kill using radiation. For example, when you have cancer, you can use a really cool exam that's called CT, PET scan. And it's really expensive to do this right now, but we hope that in the future it's going to be better. Now, the CT scan is just a regular computerized tomography examination that will see the actual shape of your insides with contrast and stuff. But the PET scan will actually measure with the electron beam transmission which area of your body is emitting radioactive isotopes. Now, if you literally drink a contrast material that has radioactive isotopes in it, and of course, this doesn't have to be bad radioactive isotopes. It could be like something like carbon-14 that's inside of you, and it happens naturally anyways, but it's just going to have something to be special that's not normally in the body. And it's a little radioactive, but not to the point that's actually going to hurt you over a long period of time, and that it's going to be metabolized out of your body before it actually does any kind of damage. There's several atoms out there which don't do very bad radioactive decays. They're tiny, and they're just emitting a little bit of radiation, nothing that's actually going to hurt you. We use them in things like fire detectors. We use them in all sorts of applications. And in fact, you probably touch radioactive stuff all the time, because a lot of the elements that show up in the periodic table have radioactive isotopes, but that doesn't necessarily mean they have bad radioactive isotopes. It's only the large ones that undergo that large decay that we talked about in the videos that become the problem. Now, in this CT-PT scan, you're actually going to use that radioactive dye to drink up. Now, if you tag with that dye on a molecule that's used by our cells, the more radioactive it is, it's going to go straight to the actual cells which are very active, because the cells which are active or have high metabolic rates will actually suck up those chemicals and be the first ones to eat them up, because they're hungry, because they're always replicating, like cancer cells, for example. So those will be the first cells to absorb the nutrient that's tagged with the radioactive thing. And so you use that to see exactly where in the body the radioactive decay is actually taking place, and therefore where the tumor is. And so the CT scan will tell you that there's something that shouldn't be there, and the PET scan will tell you if it's high metabolically active. Like that, for example, which tells us it may be an actual tumor. Now, if it is a tumor, then you can actually remove that with surgery, or you do therapy, or you do something like that. But a very cool type of surgery is something that we call gamma knife. Now, the gamma knife is basically gamma rays which are emitted by a radioactive isotope, and you basically have these tiny little gamma rays which kind of are harmless. They're very, very weak, each one of them. Even though they're gamma rays, they're not very intense and they're not very strong. But as they converge in one particular location, that location is actually going to get burned, because it's actually being focused. All the beams are being focused there, and that's actually going to cause it to be burned. And that is using radiation for treatment purposes, and it's what we call radiation therapy. And you also have other uses for this. In biology, we use it to tag molecules to see where in the cell the molecules is being active, which cells have these molecules, to do karyotypes like the one you see in the bottom right. And we also tag animals in the wild with them. Remember, these are harmless isotopes which last a short period of time. But let's say you're trying to count animals in the wild. How do you know you're not counting the same one you already counted? Now, you can go ahead and put a tag on the animal, which is going to bother him. Or you can put a small, tiny little thing that injects the animal with a radioactive isotope, something that's going to be metabolized just like the one in the picture. That's not going to last a very long time, but it will last long enough while your study is being conducted, so that if you get this animal again and you scan him, you will see that he's already been tagged because he's still radioactive. And so you don't count them again. And then you can make sure that you're counting only the ones you haven't counted. So we call that ecological tagging, right? Radioactive decay. And we can also use that to sterilize stuff. Since radiation is very dangerous and kills cells, if we want to kill certain cells, like bacteria cells, or even viruses, which are not even cells, just molecules, and we want to toast stuff, we can blast it with radiation. And so it's a method to sterilize stuff, and you see one of those radioactive sterilization ovens there in the middle there. Now, there are other uses for this as well. By using the whole half-life concept that we talked about earlier, since the half-life of carbon is 5,730 years, you can actually use this to actually predict how long something has been dead for. Now, you see, solar radiation will actually interact with the Earth's atmosphere and collide with atoms and make them actually undergo radioactive decay. And when they go into radioactive decay, remember, sometimes they expel things from the atom, and sometimes it will actually create high energetic neutrons. Now, these neutrons will be flying around the atmosphere and will collide with a nitrogen-14 atom, and you're going to have the opposite of what happens during the decay of carbon-14 that we talked before, and you're actually going to build a carbon-14 from the nitrogen-14. And so you're actually going to end up creating carbon-14 in the atmosphere from some of the nitrogen-14 that's there already. Remember, there's a lot of nitrogen in the atmosphere. So cosmic radiation is constantly creating these high energy neutrons from the breakdown of other atoms, which then hit the nitrogen-14 atom, causing it to undergo a decay and actually become a carbon-14 atom. Now, this carbon-14 atom is going to become part of things like carbon dioxide and things like that, which will be sucked up during photosynthesis by plants and then integrate into the food chain. So that means that the food chain is going to be full of radioactive carbon. Now, this carbon will decay over time during the same process we described. All right? But it's constantly being made in the atmosphere over and over again. And although it has fluctuated over time, it has on average about this much percentage of the atmosphere is made of carbon-14 right there. Okay? So you know that this is how much carbon-14 you're supposed to have. Now, since this carbon-14 is constantly being made, the atmosphere will constantly have exactly that amount of carbon-14 in it, and so will the animals, so will the plants, so will everything on Earth that's alive, breathing, and eating plants, or the plants themselves which are doing photosynthesis. Now, after you die, though, you stop consuming the carbon-14 that's going to be around, and so you're no longer going to be adding carbon-14 to the body that's now a carcass, which means that as you become a carcass, you're no longer going to increase the number of carbon-14. It's only going to decrease as it decays. So if you find like a body and you see that it has about half the carbon-14 that it should have, in other words, it only has about 300 some on this number here, okay? So it will have 300 and change for that. And so it will have half of what's supposed to have. Now you should know what that means. If it had half of what's supposed to have, it means one half-life has gone by. So you know that this specimen has been dead for 5,730 years. Now what if you find a quarter of what it should have? Then you know it's been dead for twice that, 11,460 years. If you find only 1/8 of what it's supposed to have, you know it's been dead for 17,190 years. So using methods like these, we've been able to date archaeological things and other remains of things over thousands of years. The only problem, of course, is that the amount of carbon-14 on anything is already very small to begin with. So by the time you hit more than 3 half-lives, the number is beginning to be so small that it's going to be very, very unlikely for you to be able to detect the presence of it at all. And so anything beyond about 18 to 19 to 20,000 years becomes very, very inaccurate for you to actually measure and use this. So we only use this to about the first 20 to 25,000 years, with the degree of error increasing as you go backwards in time. But it's pretty accurate over less than 17,000 years, which is as old as the human civilizations have been. So it's a very cool way to learn about the history of the Earth, but it's not such a good way to learn about geological time, because if you learn anything when you do the geological time scale project, it takes a very long time for anything to happen in geology. We're talking millions of years. And so in that case, we're going to need something that takes millions of years to decay instead of thousands of years to decay. So a good example would be uranium-238. Now, the cool thing about uranium-238 is that when rocks form at the mid-ocean ridges and things like that, or out of volcanoes, they do have uranium on them, and they also may have lead on them. But the cool thing is that because of fractional crystallization, the uranium has a boiling point that is higher, and therefore it will actually crystallize first when a rock is actually solidifying. And which means the rock, as it solidifies from the magma, it will solidify first, which means the rock is going to be made mostly of uranium. Now, outside of that, rock made of lead may form, but the center is going to be made of uranium. Now, when you study that center, and you see that it has a lot of uranium on it, you know this rock is brand new because it just formed because it's full of uranium. But if the center is suddenly full of lead, you know that this rock must be older because a lot of the uranium that was originally there has already decayed all the way to lead. And depending on how much lead is around, you know, am I in the early period of the curve or the late period of the curve? So look at that, for example. If you see that about half of what's supposed to be there is there, you know that about 4,500 million years have gone by. This is how long it takes for this complete thing to actually happen. And so if you get something like only a quarter, you know it's been billions of years. And by the way, this is already billions of years. This is 4,500 million years, so that's 4.5 billion years. So all the rocks that form when the Earth first formed are now starting to become 100% half-lifed in lead. And so most rocks on Earth will be somewhere in the beginning of the curve, around here. So because uranium takes so long to actually have this half-life, you can actually use this to date any rock on Earth. And that's pretty cool. And that's how we actually know how old Earth rocks actually are.